In this video, I want to talk about the microscopic anatomy of the bone. The primary component of the bone is bone connective tissue or osseous tissue. Bone is composed of both cells and extracellular matrix, like all connective tissue in our body. Now, I want to describe the cell and matrix that composed of the bone connective tissue, how the matrix is formed and reabsorbed, and the two microscopic arrangement of the bone. First, about the cells. We have four cell types in the bone. One of them is called osteoprogenitor, one of them is called osteoblast, one of them is osteocyte, and the other one is osteoclast. The first cell of the bone is called osteoprogenitor cell. They are stem cells which are derived from mesenchyma. When they divide by the mitotic division, another stem cell is produced along with a committed cell that mature to become osteoblast. As mentioned in the stem cells before, they are located in the periosteum and endosteum. So the stem cells which derive from mesenchyma do mitotic division, they make osteoprogenitor, and then osteoprogenitor mature to become osteoblast. The next cell is osteoblast. The osteoblasts are formed from osteoprogenitor stem cell. Often, the osteoblasts are positioned side by side on the bone surface. We don't have osteoblasts inside the bone. They are only located on the surface of the bone. Active osteoblasts exhibit a somewhat cuboidal shape and have abundant rough endoplasmic reticulum and Golgi apparatus. And by these structures, they can make matrix, which we call it osteoid. So the osteoid or matrix of the bone is synthesized and secreted by the osteoblast. Then on the surface of osteoid, we have deposition of calcium ion, which we call it calcification, and our osteoid become hard, and it can make a matrix of the bone. So the osteoblast can make osteoid. Osteoid is very soft, and then we need to have calcification on the osteoid to make a hard matrix of the bone. When the matrix becomes hard, the osteoblast trapped in the hard matrix, and then we call them osteocyte. The osteocytes are mature bone cells derived from osteoblast. They detect stress on the surface of the bone, and they can trigger new bone formation. Now look at this picture. In this picture, you can see this line, the cell line. First, we have osteoprogenitor cell. This osteoprogenitor cell develop and mature to osteoblast. The osteoblasts are immature cells in the bone, and they can make matrix of the bone. Then they trap in the matrix and they become osteocyte. So they come from each other. First we have osteoprogenitor, then we have osteoblast, and osteoblast can make osteocyte. The other cell which we have in the bone is called osteoclast. Here you can see the osteoclast. The osteoclast come from the other cell lineage. The osteoclast come from the monocyte which we have in our blood. In our blood, we have a cell which we call it monocyte. The monocyte come out of the bloodstream, go to the bone, and the name is changed to osteoclast. As you see here, the cell is very large. We have many nuclei in the cell. And this cell is like macrophages. It can do phagocytosis. And on the surface of these osteoclasts, you can see ruffled border. You have many folds on the border. We call it ruffled border. This ruffled border wants to increase the surface area, and they can do reabsorption of the bone. The osteoclast is located in the periosteum and also endosteum. They are located in a depression, and this depression has a name which we call it resorption lacuna, or the other name is Hoship's lacuna. So we call it reabsorption or Hoship's lacuna. The function of osteoclast is reabsorption of the bone. They can eat the bone and they can release calcium ion. So these are the four cells which we have in the bone: osteoprogenitor, osteoblast, and osteoclast are derived from each other, but the osteoclast is a separate bone in the separate cell in the bone, and it comes from the monocyte of the blood. In the bone, we have organic and inorganic matrix. The organic component is the osteoid, which is produced by osteoblast. In the osteoid, we have many collagen fibers, and we have semi-solid ground substance, and in the ground substance, we have protoglycan and glycoprotein. And you need to know what is the differences between protoglycan and glycoprotein. Protoglycan contain more carbohydrate, and glycoprotein contain more protein, but both of them contain protein and carbohydrate, but the ratio of protein and carbohydrate are different. This organic matrix can give bone tensile strength by resisting stretching, and it can contribute to bone flexibility. The other part of the matrix is called inorganic component. In the inorganic part, we have salt crystal, and most of these crystals are calcium phosphate crystal. The calcium phosphate interact with calcium hydroxide, and they can make an important crystal in the bone, which we call it hydroxyapatite crystal. This hydroxyapatite crystal is one of the hardest material which we have in our bone, and it can make rigidity of the bone. And besides this hydroxyapatite, we have calcium carbonate, sodium, magnesium, fluoride, and sulfate in bone too. These crystals deposit around the collagen fibers. It can harden the matrix, and it can make rigidity of our bone. So in the bone, we have two types of matrix: organic, which is semi-solid and it's flexible, and inorganic matrix, which is the hardened matrix. What about bone formation? The bone formation begins when osteoblasts secrete osteoid. The osteoid is soft. After that, on this soft osteoid, we have a process which we call it calcification. Calcification means deposition of hydroxyapatite crystal on the osteoid. Calcium and phosphate ion participate out, form crystal. And this process requires vitamin D. Vitamin D can enhance calcium absorption from our gastrointestinal tract or digestive system. Vitamin C, because we need vitamin C for making collagen, and we need calcium and phosphate for the process of calcification. So this is about bone formation. First, we have secretion of osteoid. The osteoid is secreted from the osteoblast, it's hard to write. And then the osteoblasts are trapped in the matrix and they make osteocyte. And then calcification take place on this osteoid. The other process which we have in the bone is called bone resorption. The bone resorption is a process whereby bone matrix is destroyed by substances released from our osteoclast. So osteoclast start to release some proteolytic enzyme. The proteolytic enzyme are located in the lysosome inside the osteoclast. They release it on the surface of the bone and they start to digest the organic component of the matrix. And also we have a release of HCl or hydrochloric acid. The hydrochloric acid can dissolve the mineral part of the matrix. So the organic part is broken down by proteolytic enzyme, and hydrochloric acid can dissolve the inorganic part. And then the calcium and phosphate become free and they enter the blood stream. Bone resorption may occur when blood calcium levels are low. Now about the compact bone. What do we have in compact bone? Our compact bone and the spongy bone, both of them have a unique microscopic architecture. First, about the compact. The compact bone is composed of a small cylindrical structure, which we call them osteon. The other name of osteon is Haversian system. The Haversian system is the basic functional and structural unit of a mature compact bone. These osteons are oriented parallel to the diaphysis of the bone, and they are very similar to bullseye target. Look at here. In this picture, you can see the osteon. They are, as you see here, they are very similar to the cross section of the onion. They have one central canal and some concentric layers of collagen fibers. And they are cylindrical in their shape, and they are located and oriented parallel to the long axis of our bone. In each osteon or Haversian system, you can see the central canal. This is the cylindrical canal at center of osteon, and parallel to it, the blood vessels and nerves extend through this canal, and you can see the blood vessels and nerves here. And around it, you can see these concentric lamellae or concentric layers. They are rings of bone connective tissue which surround the central canal. They are mainly composed of collagen fibers. And they can give the bone a strength and resilience. The other part which we have in the bone is called osteocyte. These cells are called osteocyte, and I have the picture of osteocyte here. Look at this picture. Here you can see the osteon or Haversian system. You can see the central canal, the blood vessels and nerves, and the concentric lamellae which we have around the central canal. Between these concentric lamellae, you can see a star-shaped cell. These star-shaped cells are called osteocytes. Actually, they are the mature osteoblasts which are trapped in the hard matrix, and their name changed to osteocyte. The osteocytes are mature cells. They are non-producing cell, but they are live cell. And you can see them in the inside, the bone. The lacuna are these small space which the osteocyte is located. In each lacuna, you can see one osteocyte. And our osteocyte is a star shape. It contains many processes, and these processes are located in tiny interconnecting channels inside the bone. These tiny channels are called canaliculi. The canaliculi house the osteocytic projection. And these osteocytic projection from this cell and the neighboring cell can make gap junctions together. So the oxygen and nutrient can pass through the central canal to the first osteocyte. By the gap junction, it can go to the next osteocyte and go to all the osteocytes which we have in one osteon. And also the waste product and carbon dioxide can go to the opposite direction, cell to cell, to reach the veins here. So this is the structure which we have in our compact bone, and this osteon is the functional unit of our compact bone. Now I want to show you a large piece of compact bone. Look at here. In this compact bone, you have different parts. The first part which you can see here are these osteons. All of them are like the cross section of onion, and you see that their longitudinal axis is parallel to the long axis of the bone. They have a central canal, each of them, and in the central canal you can see the blood vessel, the like arteries and veins, and the nerve. Between these osteons, you can see some bony parts. These bony parts are called interstitial lamellae or interstitial system. The interstitial system are either the component of compact bone that are between osteon or the leftover part of osteon that have been partially reabsorbed. Thus, they often look like a bite has been taken out of them. The interstitial lamellae are incomplete and typically don't have any central canal. The other parts which we have here are inner circumferential and outer circumferential lamellae, or internal and external circumferential lamellae. They are ring of bone immediately internal to the periosteum of the bone or immediately external to the endosteum. The one which is located near the periosteum is called external or outer circumferential, and the other one which is adjacent to endosteum is called internal or inner circumferential. And they can protect the bone from outside, and also they can protect the marrow cavity from inside. So these are the different parts which we have in our compact bone. You can see osteon. Between osteon you have interstitial lamellae. Around the bone you have external circumferential, and around the marrow cavity you have internal circumferential lamellae. Here you can see the location of endosteum, and out of the bone you can see periosteum. And as you know, the periosteum has two layers. The fibrous layer is located outside, and the cellular layer is located inside. The other part which I want to show you in this picture are these perforating fibers. The other name of these perforating fibers or Sharpie's fiber, and I told you that they can connect the periosteum to the bone. And here in this picture, you can see some canal which can attach the central canal together. We call them perforating canal or Volkmann's canal. They are resemble central canal in that they also contain blood vessels and nerves. However, perforating canal run perpendicular to the central canal as you see here, and they help connection of these central canals together. The next part is about the spongy bone. In the spongy bone, we have a structure which we call it trabeculi. Unlike the compact bone, the spongy bone doesn't have any osteon. Instead, they have the structure which is an open lattice of narrow rods and plate of the bone. Look at here. You can see these plate of the bone. They are very narrow and they don't have central canal, so we don't call them osteon. This structure is called trabeculi, the trabeculi of the spongy bone. The bone marrow is located between these trabeculi. This spongy bone can make resistance to different stress. We have osteocyte in each trabeculi, and each trabeculi is covered by endosteum, and in endosteum you can see osteoblast and osteoclast. So osteoblast and osteoclast are located around on the surface, and inside the spongy bone you can see osteocyte. The next part is about the hyaline cartilage. The hyaline cartilage is a connective tissue, and it contains cells and matrix. The cells are scattered through matrix of protein fibers, and we have a gel-like ground substance in the cartilage. And in this ground substance, we have protoglycan, and we don't have any calcium ion. So the cartilage is semi-solid and it's not as rigid as the bone. It's resilient and flexible. It has high percentage of water. It's highly compressible and a good shock absorbent. The cartilage is avascular and it doesn't have any blood vessel. And we have two types of cells in the cartilages. One of them is called chondroblast, which can produce matrix of the cartilage, and the other one is called chondrocyte. The chondrocytes are located inside the matrix in the spaces which we call them lacuna, and they can maintain the matrix. And around the cartilage, we have perichondrium, which is the dense, irregular connective tissue, cover cartilage and help maintaining the shape of the cartilage. Now you can answer these questions. What are the functions of the osteoprogenitor cell, osteoblast, osteocyte, and osteoclast? What organic and inorganic substances we have in the bone matrix? What are the major components of the osteon? And what are the primary ways that hyaline cartilage tissue differ from the bone tissue? In this part, I want to talk about the growth of the cartilage. The process of growth cartilage are different, and it begins during embryonic development. And we have two types of growth. One of them is growth in length, which we call it interstitial growth. One of them is growth in width, which we call it appositional growth. First, I want to talk about interstitial growth of the cartilage. Look at here. In this picture, you can see the cartilage. The cartilage cells are chondrocyte, which are located in the center of the cartilage, and chondroblast, which are located around. And here you can see the perichondria. What happened in the interstitial growth? The interstitial growth happened inside the cartilage. Here you can see a chondrocyte, and the chondrocyte is located in this cavity, which we call it lacuna. And this chondrocyte start to do mitotic division. The result of mitotic division is two daughter cells. These two daughter cells are young, so we call them chondroblast. And you see that they are located in lacuna. They are located in one lacuna. Then each of these chondroblasts start to make matrix. When they make matrix, they separate from each other. And as you see here, each of them is located in a separate lacuna. And now we call them chondrocyte. And you see that inside the cartilage, first we have one cell, then they become two, they go far from each other. And we have growth inside the cartilage. The cartilage continue to grow internally, and we call it interstitial growth. The other type of growth which happen in the cartilage is called appositional growth. And in appositional growth, we have growth in the surface of the cartilage. Look at here, this is the surface, and appositional growth happen on the surface. Now, I want to go a step by step. We have mitotic division in the stem cells, which we have in the perichondrium. In the perichondrium, we have some mesenchymal cells, which are stem cells. They do mitotic division, and by mitotic division, they make new cells. And then these new cells differentiate to chondroblast. The chondroblast start to make matrix, as you see here. And when they make matrix, they become separated from each other, and they can increase the girth of the cartilage, or they can increase the diameter of the cartilage, or they can grow our cartilage can grow externally. So these are the two different processes of growth. Sometimes we have growth from inside the cartilage. Sometimes we have growth from outside of the cartilage. We call them interstitial and appositional growth. And now you can answer this question. Where do interstitial and appositional growth of cartilage occur? And the last part of this video is about fracture repair and what happens when we have a fracture. Look at here. I start from phase 1. In the phase 1, we have a fracture hematoma formation. A bone fracture tears the blood vessels here, and the blood vessels are broken down in the bone and also in the periosteum and causing bleeding. This bleeding results in a fracture hematoma that forms from the clotted blood. After that, we have a step 2, which is a fibrocartilaginous callus formation. Regenerated blood capillaries infiltrate the fracture hematoma. First, the fracture hematoma is recognized into an actively growing connective tissue, which we call it procallus. Fibroblast within the procallus start production of collagen fibers that help connect the broken end of the bone. The chondroblast in the newly growing connective tissue form a dense regular connective tissue associated with the cartilage. Eventually, the procallus become a fibrocartilaginous callus, which is a soft callus. And this fibrocartilaginous callus stage lasts at least 3 weeks. Then we have a hard or bony callus formation. Within a week after the injury, osteoprogenitor cells from the area which are near to the fibrocartilaginous callus become osteoblast, and osteoblast migrate here, start to make the new bone. The fibrocartilaginous callus is then replaced by the bone, which form hard or bony callus. The trabeculi of the hard callus continue to grow and thicken for several months. And then we have a remodeling of the bone. Remodeling is the final phase of a fracture repair. The hard callus persists for at least 3 to 4 months as osteoclasts remove excess bony material. Look at here, first we have bone here, but the osteoclast come and remove all of this bony material. Compact bone replaces primary bone, and the fracture usually leaves a slight thickening of a bone, as we can indicate it by the X-ray. However, in some instances, healing occurs with no persistent obvious thickening. So in the fracture repair, first we have hematoma, then we have callus formation. Here the connective tissue come, and this connective tissue is a cartilage, and then we have migration of osteoprogenitor cells and osteoblasts. They make bone, and after that we have remodeling. The bones which are located in the bone marrow are reabsorbed, and the bones which we have around in the diaphysis become harder and harder, and we have little thickening, which after remodeling and after some months all of them are gone. And now you can answer these questions. What are the 4 basic steps in the fracture repair, and explain the risk of bearing weight on a bone when a fibrocartilage cartilage callus is forming. Think about this question, and I will talk about it in the next video for you.